everyone, I am Twoot, and in this video, we will be talking about the TGF-BETA signaling pathway. The TGF-BETA or transforming growth factor beta pathway is a very complex pathway involved in cell differentiation, embryogenesis, organogenesis, homeostasis, apoptosis, and many other cellular processes. Now let's look at the ligands of the TGF-BETA pathway. The ligands of the TGF-BETA superfamily include TGF-betas, including TGF-beta 1, TGF-beta 2, and TGF-beta 3. Another one of the ligands are the BMPs, also known as the bone morphogenetic proteins, GDFs, the growth and differentiation factors, activins, activin A, B, and AB, the AMH, called the anti-malarian hormone, and nodal. Now let's look at the receptors of this pathway. The TGF-betas 1, 2, and 3 bind to the TGF-beta receptor type 2 and are involved in cellular processes such as embryogenesis, cell differentiation, and apoptosis. The BMPs bind to the BMPR2 receptor and are involved in osteogenesis, cell differentiation, anterior-posterior axis specification, growth, and homeostasis. Activins bind to the ACVR2A receptor and are involved in processes such as embryogenesis, osteogenesis, and hormone regulation. Now let's talk about SMADs. SMADs are intracellular proteins that transduce extracellular signals from TGF-beta pathway ligands to the nucleus, where they activate downstream gene transcription. They are between 400 and 500 amino acids long. They consist of two globular regions at the amino and carboxy termini connected by a linker region. These globular regions are called methomology 1 or MH1 at the N-terminus and methomology 2 or MH2 at the C-terminus. The MH1 domain is primarily involved in DNA binding, while the MH2 is responsible for the interaction with other SMADs and for the recognition of transcriptional coactivators and co-repressors. There are three types of SMADs. The receptor-regulated SMAD, or the R-SMADs, consist of SMAD 1, 2, 3, 5, 8, and 9. They are involved in direct signaling from TGF-beta receptor. Co-mediated SMAD, or co-SMAD, is SMAD 4, which interacts with the R-SMADs to form heterotrimeric complexes and then act as a transcriptional factor. Inhibitory SMADs, or the I-SMAD, are SMAD 6 and 7. Their function is to suppress the activity of R-SMADs. TGF-betas, activins, nodals, and some GDFs are mediated by SMAD 2 and 3, while BMPs, AMH, and a few GDFs are mediated by SMAD 1, 5, and 9. SMAD anchor for receptor activation, or SARA, facilitates the process of recognition of R-SMADs by the receptors and their efficient recruitment and phosphorylation. Next. What, huh?
So many things to remember.
Moving on, we're gonna be discussing some pathways now. The pathway we will discuss involves two receptors, type 1 receptor, type 2 receptor, and the signaling molecule TGF-beta. Upon receiving the signal of TGF-beta, the type 2 receptor monomers come in proximity and dimerization takes place. This further leads to the recruitment of type 1 receptors, and together they form a heterotetrameric complex. Type 2 receptor phosphorylates type 1 at serine residues due to its kinase activity. Now, with the help of a protein called SARA, which is anchored at the cell membrane, SMAD 2 and 3 are recruited to the receptor. SMAD 2 and 3 are also phosphorylated by type 1 receptor. This phosphorylation of SMAD 2 and 3 is followed by a conformational change in the MH2 domain of SMADs, which then leads to its dissociation from the receptor complex as well as SARA, after which they bind to SMAD 4, which is a co-SMAD. This heterotrimer complex is then translocated to the nucleus, where it binds to the transcription promoters and cofactors, causing DNA transcription, making proteins involved in regulation of apoptosis, immunosuppression, and so on. Let's discuss the involvement of this pathway in cellular processes and cell cycle control. TGF-beta, being a multifunctional cytokine, plays a very important role in many different processes, including cell cycle control, regulation of development, differentiation, ECM formation, angiogenesis, chemotaxis, and induction of apoptosis. TGF-beta pathway gets activated by binding of ligand to the receptor, which causes its dimerization followed by phosphorylation of type 1 receptor by type 2. This process leads to the activation and phosphorylation of RSMAD. RSMAD then binds to a cosMAD forming an oligomer and moves to the nucleus. This leads to the transcription of some target genes, which eventually results in an increase in cyclin inhibitors. In case this inhibition is not done, cell cycle is not regulated, which can lead to cancer. Now that we're done with the pathway, let's...
Please tell me we're almost done.
Almost. Well, anyway, TGF-beta 2 receptor mutation is a germline mutation. Environmental factors such as chemicals, race, etc. can lead to mutation. Let's see what happens when mutations occur. In case there is a mutation at receptor 2, no attachment to ligand or dimerization takes place. Thus, SARA and SMAD will not bind to receptor, which of course means that there is no activation of cosMAD or transcription of target gene. As a result of these events, cell proliferation occurs, apoptosis decreases, cell cycle regulation is disturbed, consequently, tumorigenesis results. This then results to colorectal cancer, which is the third leading cancer by incidence. It can be caused due to mutations in receptor 2 gene or in SMAD proteins, making it a multifactorial disease.
Oh no, I don't want cancer.
Time to discuss diseases.
Uh-oh.
This pathway can result to autoimmunity. When TGF-beta binds to interleukin 6, it results to the proliferation of T helper 17 cells, which have been linked to inflammation and autoimmunity. Thus, TGF-beta could have a role in causing autoimmunity. While TGF-beta suppresses tumor growth and cancer in early phase of neoplasia, it can promote tumor progression and metastasis in later phases. The cancer cells in the body also produce TGF-beta. This TGF-beta can lead to metastasis. And that's all we have for you about the TGF-beta pathway. Thank you for watching.